Okay, we'll go ahead and get started. So again, good morning, everyone. Welcome to today's webinar. My name's Amanda Miller. I'm the Client Operations Director here at Inorganic Ventures. So we're very excited to have everyone here to introduce you not only to Inorganic Ventures, but also to Seal Analytical. Today, we're going to be presenting on the Seal Analytical ready to use discrete analyzer reagents. We'll also share some performance studies with everyone and also just go through some specific application use purposes. So with that said, I'm going to go ahead and kick it off and let everyone on the team introduce themselves. And then Liv, if you want to go ahead and share your screen for the presentation, Joey's going to start it. So we'll just go down the line. We'll have him introduce himself last since he's going to kick off the presentation. Also, if you guys have any questions, please submit them in the Q&A box. Just type them in there. I'll be moderating those, and then we'll go through the questions at the end of the presentation and answer all of those at that time. So without further ado, I'll let Liv Forbes go ahead and kick off by introducing herself, and then we'll go to Johan, and then last but not least, Joey, who will start the presentation. Thanks, guys.
Hey, everybody. So my name's Liv Forbes, like Amanda said. I've been with Inorganic Ventures since 2019. I'm a research and development chemist here. I've done a lot of work in partnership with Seal to develop these reagents, and we're really excited to share with you guys and introduce them. And I'll kick it to Johan.
Hey, everybody. I'm Johan. I've been at Seal for getting close to 9 years now. I'm the marketing manager now, but I started as a technical support chemist and then a technical sales chemist. So I know my way around the laboratory and the reagents and the methods as well. Excited to be here and working with Inorganic Ventures on this very exciting project.
Okay, that leaves me. Good morning. My name is Joey Redovich. I am the technical sales and product manager here at Seal. I started in 2010, which, if you can do some math, was a while ago now. So what we're going to be doing today, or at least what I've been setting up to talk through, are our discrete analyzers in general. So I'm going to make sure we get this working. So everybody can see those slides now? All right, excellent. I did it correct. I always get a little nervous. We do webinars on a different program. So Seal as a company, we are based in Macon, Wisconsin, as far as our US group goes. So that's where Johan and I are today. Here we manufacture our discrete analyzers and our digestion hardware. So we have a TKN block for digestions. We also have an automated metals digestion block. The US is probably our biggest office. However, we are actually owned and operated through the UK. So we're owned by a company called Port Vale in the UK, and then we have a support office in the UK as well. We do manufacturing in Germany and the Netherlands. While those are not our discrete analyzers, we make segmented flow systems, which automate the same chemistries that discrete analyzers would automate for you. And then in our Netherlands facility, we manufacture robots. So those are going to be, not the robots that are going to be walking around and bringing you stuff, but robots that are going to be running BOD or automating tests such as, pH, conductivity, alkalinity, turbidity, stuff like that. And then we also have a support office in Shanghai to help support all of our Asian facilities. We don't make anything there, but we do, again, where our three main manufacturing sites are in the US, the Netherlands, and Germany, with the US being where we manufacture the discrete analyzers we're going to be talking about today. So what is a discrete analyzer? Well, odds are if you came to a webinar about discrete analyzer reagents, you probably have a pretty good idea. But just in case, a discrete analyzer is going to fully automate the wet chemistry process. So it's going to take samples, take reagents, and put them into a well. The name discrete analyzer comes because each test is actually done in its own discrete well. While it's automating the different methods that we may be running, whether it be ammonia or phosphate or nitrate, it's also able to do a number of additional automation things for us. That includes an auto calibration curve. So we give it a single top standard. It will make dilutions of that top standard generate a calibration curve. It will dilute overrange samples. It will automatically spike samples if we ask it to, and it can have a wide array of QCs being inserted, whether they be your midpoints, your blanks, ICVs, spikes, duplicates, all sorts of QC insertion that we would need. And with that comes a variety of rules that we can use should one fail. We can assign rules for the system and go back and rerun samples and then bracket with those passing QC. Again, a discrete analyzer is essentially designed to, I put my samples on, I put my reagents on, I hit go, I go have a coffee or do other things throughout the lab. So the first step is the sample and the reagents are going to be mixed. So there's a quick little video of how our 400 is working. So the sample arm is going to use that syringe. It's going to pick up our sample. It's going to pick up our reagent, and it's going to place it into one of those small wells. After it does that, we're going to allow some time for incubation. So the sample and the reagent are going to sit in that well. That reaction ring is heated to about 40 degrees C. It's going to help catalyze the reaction as soon as that reaction time has come to a completion. Now that's preset depending on the method that we're running. The sample is going to be pulled into our 1 centimeter flow-through cuvette. It's it's quartz glass, so it's as optically pure as we can get. It allows us to see very clearly and help to push our detection limits low. So that absorbance is red, and then we take those absorbances and we generate a calibration curve. So we draw a line of best fit using the target concentration and the absorbance of the sample. We then will read sample absorbances and plot them against this curve. Right? All in all, it's a relatively simple process. However, the automation and allowing the system to do it instead of a chemist needing to do it in the lab is pretty handy, especially if you have a lot of samples and a lot of different methods you're trying to automate. A lot of people think that discrete analyzers are a new phenomenon. However, it was actually invented in 1959, which is even longer than I've been at SEAL. So that's good. I'm not quite as old as discrete analyzers. So this was the Robot Chemist, which launched in 1959. It's a little before SEAL got into the game. So in 1958, Technicon invented the segmented flow analyzer, which is the auto analyzer, which today is now known as the AA500. So that's the latest version. So we were doing colorimetric testing way back then. However, we didn't start making discrete analyzers until the 80s. So the Aqua 800 was something that we launched in before SEAL was SEAL. So it used to be a company called Cinermed. And this was mostly found in Europe. And then we brought the AQ2 to life in about 2000. And shortly thereafter, it came to the US. AQ400 was launched in 2015. And then our big boy, the AQ700, was launched in 2022. Not shown on here is our AQ1, which we did in 2010, and our AQ300, which is in 2017. Doesn't mean we don't like them. They're just, again, these are our larger models that are actually on this slide. A little history of how SEAL did work with discrete analysis. So again, I mentioned in 1986, Cinermed launched the first large-scale discrete analyzer. So the name SEAL actually comes from Cinermed Environmental Analytical Laboratories. So SEAL used to be an acronym. Now it's just SEAL. So we're not part of Cinermed anymore. To be honest, I don't even know if Cinermed is a company. Maybe they are, maybe they aren't. But SEAL then launched in 2002, in 2000 AQ2, and then in 2002 we moved to the US. So SEAL US was founded in 2002. From 2002 through 2005, we worked steadily with the EPA to get various methods approved on the discrete analyzer, right? No one had seen one before. It was kind of a weird new automated technology. So the team at SEAL worked with the EPA to get that approved. And then in 2005, the EPA Region 4 did recognize the discrete analyzer as a approved option for automating this type of testing. From 2005 to 2007, then they required us to actually send method data into the EPA for the method to become approved. So we had to have individual EPA letters of approval for any method that we wanted to run. But then in 2007, pretty sure they just got sick of us sending a bunch of methods to them to look at all the time. They released a document called flexibility to modify. And what that means is as long as we are holding to how the chemistry is in the EPA method or the standard method, um, we're not making any dramatic changes, and those changes that are allowed are actually listed in this document. Our method's automatically approved, right? We don't have to go through the process of running it over and over and over again and sending all that data to the EPA and then have them run the method to make sure that it's approved. It just is. So from there, AQ2 became a very popular system. We sold hundreds of them. In fact, the oldest AQ2 that was installed in 2002 is still running today. Now, that doesn't mean all of your AQ2s, all your discrete analyzers are going to run for 23 years. You have to take good care of them. But that's a pretty interesting fact, I think. And then again, how we launched our later systems in 15 and 17 came the 400 and 300. And then in 2021, 2022, we launched that AQ700. A couple of things that we like to bring up while we are looking at how discrete analyzers operate is first at the detector. All right, what type of cuvette is your instrument using? I mentioned earlier, we are using a optically pure quartz glass cuvette. Every sample is read in that same cuvette in the same orientation. That allows us to get the reproducibility that we want and allows us to, again, push our detection limits as low as we possibly can because we're not fighting different optical purities of plastic cuvettes. When we're mixing, right, so when we're taking these teeny tiny volumes, our total reaction volume for each test is somewhere between 500 and 700, 500 and 800 microliters, right? So we're talking really, really small volumes. We need to make sure that we're accurately mixing as well. So rather than doing any like vibration or shaking or anything like that, what we're going to be doing is we're going to put our sample probe in, we're going to be aspirating everything up, and we're pushing it back out, aspirating everything up and pushing it back out. And that's going to cause everything to mix very thoroughly in our small wells. Speaking of those small volumes, how are we going to ensure that those small volumes are being accurate? Well, we have a 1,000 microliter syringe capable of outputting as little as 2 microliters. Okay, so we use this larger syringe, 1,000 microliters, which is a milliliter. We use that slightly larger volume to ensure that we're able to get a nice wide calibration range. And then we're also going to be able to make good sized dilutions on overrange samples to allow for a wider range of samples to be ran on a single run. And finally, washing, right? We talked about it. This is a magic little box. It's going to be able to automate 4, 5, 6, 7 different methods at a single time. So how are we ensuring that sample 1 doesn't interfere with sample 2, and also that chemistry A is not going to interfere with chemistry B. We have various rinse stations on the system that are all, essentially cross-checking each other, taking absorbance values of DI water within the cuvette. But probably the most important portion of our system is our probe wash assembly, which is pictured here. This is going to be cleaning the probe before it rotates. Okay, so when a probe goes into liquid and it comes up, what's on the tip of that probe? Usually a droplet of whatever it was just in. So if that arm is moving very quickly, that could end up in another sample, in another reaction well, in a reagent container. So what we have decided to do is we vacuum DI water through this small circle at a high rate of speed. So as the probe comes up, it works as a squeegee and cleans off that probe before it even moves. Now it's able to rotate, go pick up the next sample or pick up the next reagent without having to worry about dropping any droplets of whatever we were just in another sample. And so a couple years ago, we started this project with partnering with Inorganic Ventures. Something that has been very important to us is their shared focus on accuracy and reproducibility, and of course, compliance. Now, I'm not the compliance manager here at SEAL, so Sarah McFall always keeps me in line on that. But the way that IV goes about things with their strive for excellence has been something that's been very important to us, and one of the reasons that we wanted to work with them. We know that they have worked closely with various standards and other companies for EPA aligned, ready to use other stuff, not necessarily reagents for specifically discrete analyzers, but we saw a hole in the market where we had been getting some requests for these, but we weren't able to necessarily fulfill it. Where IV has all of the people and the facility that is prepared to handle reagent makeup. Pretty much everybody knows them, right? When I was mentioning this to various customers across the country, hey, we're trying to partner with Inorganic Ventures to get this pre-made reagent journey started. Everybody's like, oh, they're such a great company. I had met Liv several times at some shows around the country as well. And again, we were very excited to start working with the team over there. And the thing that we're bringing, of course, I think to our customers, which we're going to talk a lot about here, is the goal is to make everything easier, right? Discrete analyzers have made things easier for a long time, right? We talked about the automated dilutions, automated calibrations, automatic chemistry changeover, right? I can put samples on, run 4, 5, 6 different methods, and I don't have to be there. One of the time restraints on customers right now is being able to prep those reagents themselves. So by offering pre-made reagents through IV, we're able to cut that down as well. So it's been a journey. There was some hiccups along the way, but we are very excited to launch this. Uh-oh, I think I did it wrong. Did I stop sharing?
No.
No? How about now?
Okay, there you go.
Okay.
Perfect. Thank you for the glowing review, Joey, and the intro to SEAL. I even learned some things about you guys, so that was really cool. I'm going to talk just a little bit. Hopefully the slides are working like they're supposed to. I'm going to talk a little bit about, who IV is, and then I'm going to talk about some stability studies that we've done, and then I'm going to toss it over to Johan to talk about what reagents we have available. So for those of you who aren't familiar with Inorganic Ventures, can you guys see, are your faces on my slides? Okay, perfect.
We're at the top, but
That's fine.
I just see the slides.
So for those of you who don't know, Inorganic Ventures is a manufacturer of inorganic certified reference materials. We've been in business for over 35 years. We offer a variety of solutions, including high purity single element standards, custom blends, pH standards, titrants, and what we're talking about today, reagents. We're ISO 9001, 17025, and 17034 accredited. And because of our accreditations, we're required to have teams to do, pretty robust testing on everything that we do, including a really involved stability program, which is something that I've had a hand in my time here at Inorganic Ventures. So we'll talk about that a little later, but the stability studies are really what gets this reagent going. It's a big hurdle for these reagents. So we'll talk about that in a couple slides. So here at Inorganic Ventures, we want to be chemists working for chemists. We're experts in custom blends, not just reagents, but, custom standards, whatever you need, tailored to fit your method. We'll do what we can to help you design reagents or standards or titrants or whatever you need for your methods. We've got pretty personalized support. We have a couple different teams for product design, for client operations, R&D, our QC teams. If you have questions, anyone's, available to help and answer whatever genre of question you have as well, whether it's technical or about whatever you've got going on. We have QRM who can help. So, we just have a large team of people who want to be a resource. So we've got pretty fast lead times as well. For the pre-made reagents, it's actually 2 days. So within 48 hours of placing your order, you should get a shipping notification, which is pretty cool. Much faster than if you were to be ordering the dry starting materials to buy them or to make them yourselves. So that's, I think, really handy to have. And it makes getting new methods set up pretty quickly as well. Because like I said, if you want to be testing new methods, and you're going to have to order all the starting materials, and that's going to take who knows how long, depending on what vendors you're going through. Or, you can just order the reagents for the method you want to try and have them, in however long shipping takes. And we want to offer solutions not just in a literal sense, not just liquids in a bottle, but we want to be, a resource. We want to troubleshoot with our customers. And I think this project is a really good show of that. So, like Joey mentioned, making the pre-made reagents or making the reagents in-house, it's a time sink, and long-term stability is an issue with these as well. So we want to, develop options to help customers with, whatever they've got going on. So like I said, like Joey said, with the reagents, stability is an issue, long-term stability. Like I mentioned earlier, our 17034 accreditation requires us to have a really robust stability program. So we're, pretty well equipped to handle doing stability studies on different kinds of products. So these reagents are, they've been interesting to work with for sure, but, throughout the project, we've done a pretty good job at collecting some really, really good data to support, that long shelf life that we've given them. So now I'm going to talk a little bit about our stability studies and how we designed them. So we wanted to do room temp and refrigerated studies. We didn't really have a lot of faith in the room temp reagents just because of, what we've heard from users before we got into this, what we've seen ourselves. But we still did that testing every 6 months on those reagents. For the refrigerated reagents, we tested them every 3 months. And we'll continue collecting data about these reagents for as long as they're, performing well. So if, we've launched a reagent with 2 years of stability, that's still in stability and still getting testing. And we might update that to 2.5 years or 3 years, depending on, how that testing goes, because we don't want to, offer something that's not stable for the full time that we're giving it. Of course, that's not an option, but if we can generate more data and give a longer shelf life, then we're happy to do that. It makes your lives easier. So like I said, the longer we can collect data and the longer these reagents are performing well at the standard that we expect of them, then we can continue to extend those shelf lives. But as for now, I think the highest that we have is 2 years. So that's, still much better than like a week or a month that a lot of these methods list. One thing that we did want to mention, like I said, refrigeration is kind of key. Well, it definitely is key. The room temperature reagents aren't really happy. A lot of them go through color changes or have some chemical instability that, really hinders their efficacy. We also, during our studies, protected these from light, and we recommend that customers do the same. They're going to come in a TCT bag, which is transpiration control technology. For these reagents, transpiration isn't such a huge deal. For certified reference materials, it's a lot more important because that certification on that concentration of your standards is a lot more important than, the chemistry is happening with these reagents. So the transpiration isn't as much of a deal, but the protection from light is really important. So you can just fold that bag back over and just, binder clip it or paper clip it, and it'll keep your reagents protected from light while you're storing it in the fridge. We know that there's a light in there. Who knows if it turns off when you shut the door? Because we've all tried. So the bag just, makes sure that your reagents are safe. So we're going to go over a little bit of data from some of the methods that we have available. So for phosphate, this reagent's stable for 2 years, 24 months. You can see after 833 days, the reagent's still performing really, really well. The absorbances are still where they're supposed to be. There's a little bit of natural variance in everything, including the reagents and how the instrument's performing and the samples that you're running. If you're using different standards, you're going to get a little bit different results, just minor, but you can see the reagents are still performing really, really well after, quite a long time. So we're pretty confident in the data that we've collected for these. Here's ammonia as well. So you can see this is up to 461 days. Ammonia doesn't have a 2-year shelf life because it's not stable for that long. Our data doesn't support that. I believe it's got 12 or 15 months. So you can see, up to that point, it's still performing really, really well. But after this point, the data quality goes down and the reagents don't work as well. So, that's just not something we want to offer. So we limited that shelf life. And then Johan, like I mentioned, is going to talk about what reagents are available now. We're also having a total hardness reagent set coming soon. And we've got pretty good stability data for these. The only difference with the total hardness is that we've had to tweak the reagents a little bit from the seal method, just to ensure that long-term stability. So same chemistry is going on. They're just organized a little bit different in solutions so that, like I said, that long-term stability is still something we can achieve. And you can see here, after 700 days, it's still performing really, really well. So that's what we really like to see. Overall, pre-made reagents, like Joey mentioned, we want to make your lives easier. The discrete analyzer instrumentation itself already does a lot to make analysis easier, but we want to just, make it even easier if we can. So the pre-made reagents reduce the burden on technical staff. They don't have to make these reagents. It's a time savings because, the reagents aren't necessarily difficult to make, but they are time consuming, and the pre-made reagents are ready right now. As well with the pre-made reagents, like I mentioned earlier, you don't have to keep those starting materials on hand, that dry material, which is easier for your inventory management. And it's also safer. Some of these starting materials are a little bit hazardous. And it also reduces the risk of cross-contamination from your starting materials. So, making the reagents for one method might contaminate the reagents for another method. So it's just something to be aware of, and definitely some ways that we've tried to make you guys' life a little easier. So now, like I mentioned, Johan's going to talk about the reagents that we have available and also some seal-specific like method info.
Great. Thanks, Liv. 1 second while I also get used to how Zoom works. I'm not quite elegant with this platform yet. All right, are we good? We can see the slides? Excellent. Okay, so I'm just going to talk a little bit about what reagents we have available right now. They're open for ordering on the Inorganic Ventures website. We are starting with these methods, and then we're taking feedback from our customers on what methods we'd like to see next. I know TKN is a very big popular one right now, but the more requests we get for a method, the more confidence we can have in opening up that product line. So let us know what methods you're running that you'd like to see pre-made reagents for, and we can add that to the list and keep a tally. So to start, we have ammonia, one of the most popular methods. We are currently doing pre-made reagents for the ammonia methods we have for the salicylate reagent. We're not doing the phenate reagent right now. The salicylate method is nice because it's only 2 reagents, which means it runs faster on a discrete analyzer. It's also less hazardous. Phenate is a carcinogen. We like to avoid that by using the salicylate reagent instead. So this is applicable for the seal methods EPA-148 going up to 1 milligram per liter, EPA-150 going up to 5 milligrams per liter, and EPA-153 going up to 10 milligrams per liter. Each of these seal methods uses the exact same reagents. So if you have the salicylate for one, you have the salicylate for all of them. These are all following EPA 350.1 for the actual EPA method. Just so you don't get too confused by the seal method numbers. That's different than the actual EPA method numbers. They're all based on EPA 351.1. The DCI is a nice reagent as well. On the phenate method, we use bleach as the hypochlorite source that we need for the reaction. In the salicylate method, we use DCI, which is made from a dry chemical dichloroisocyanurate. It's a little bit more stable, a little bit more predictable than using like store-bought bleach, which can vary from batch to batch depending on what Clorox or Great Value or whatever brand of bleach you use decides to add into their product line at that point. That store-bought bleach isn't quite as well regulated as a nice ACS grade reagent. So we like that DCI reagent quite a bit. Next up, we have orthophosphate, another one of the big 3 methods. So our orthophosphate methods all use different ratios in the color reagent. So the pre-made reagents we have available right now are for our EPA-118 method going up to 1 milligram per liter. That's our most popular one. The one that goes up to 5 milligrams per liter, up to 12.5 milligrams per liter, all the way down to sub ppb levels with a top standard of 0.2 milligrams per liter. Those all use different color reagents. Some of them use the same ascorbic acid. So check your method document. If it's using the same one as EPA-118, it's 1.5 grams per liter, or sorry, 1.5 grams per 100 milliliters of reagent, you could still use that ascorbic acid. The ascorbic acid does not include the phosphate spike that's mentioned in some of our methods. The phosphate spike is optional. We did not include it because some regulatory bodies, such as the Wisconsin DNR, have requested that that not be added in. The reason we add it in some cases is to bump up the color just a little bit across the board evenly on every sample, every standard, every blank. By bumping up that color just a hair, we avoid a little phenomenon that can happen on the bottom of the calibration where the blank reads slightly higher than the next standard and then it goes up from there. So it makes a check mark shape. By adding in that phosphate spike and just slightly elevating everything, we avoid that from happening. So if you're having that checkmark shape issue, you can add the phosphate spike back into this pre-made ascorbic acid, but it doesn't come with it in, just so that it can be used whether you're in Wisconsin or any state. Next up, the next of the big three is nitrate plus nitrite. And this is for cadmium reduction. We also have hydrazine reduction, which is coming up next. These reagents are for the cadmium reduction method, the most popular method on sealed discrete analyzers for nitrate. This is for EPA-127 going up to 2 milligrams per liter, EPA-126 going up to 5 milligrams per liter, and EPA-114 going up to 15 milligrams per liter. These are all following the EPA method 353.2. Again, the reagents are identical no matter which one of these ranges you're running. So we have here the buffer reagent with the Triton added in, as well as the sulfanilamide NED reagent for the color development. Like I mentioned, we also have nitrate plus nitrate using the hydrazine reduction. This is for our EPA-141 method going up to 1.5 milligrams per liter. I want to say it's EPA method 353.1. But I could be mistaken there, so don't quote me on that. If you download the Inorganic Ventures reagent sheet, it'll show you what EPA method each of these is equivalent for. So this is going to include your alkaline buffer, your working hydrazine, which does the reduction step of nitrate to nitrite, and then also the sulfanilamide NED reagent. That sulfanilamide NED reagent here is different than the one used on the cadmium reduction. So make sure that you order the correct one. The bottle will say either NOx CAD or NOx hide for hydrazine. Then we have chloride, one reagent test, nice and simple, very quick on our discrete analyzers. This is a great one to buy pre-made because the reagent contains mercury. It is not fun to prepare yourself. You have to be very careful as you're, dispensing any leftover reagent. When you're making it, you want to be careful not to get it on your skin, of course. So nice to have it just pre-made in a bottle ready to go. Pour it into your wedge, start it up. That's a nice streamlined approach for chloride testing. So we have this for EPA-105 going up to 100 milligrams per liter, or EPA-124 going up to 200 milligrams per liter. Again, same reagent for both tests. And then the other big one reagent test, sulfate. This is not a colorimetric test. This is a turbidimetric test, meaning instead of developing color with the reagent, we're developing turbidity or cloudiness. And then we measure that difference between clarity and cloudiness to build the calibration curve and compare those samples to the calibration curve. So we call it the gelatin reagent. It's got some barium chloride as well as some gelatin to create a suspension. This is another one that's nice to have pre-made and tested by Inorganic Ventures, because again, that gelatin quality in the dry reagents can be difficult to ensure. Some vendors have better ones than others, and then sometimes it switches to a different vendor being better than others. I know that we've seen a lot of variance in the performance between different gelatins. And so it's really handy to have this one from Inorganic Ventures. We've started using it in our lab as well, just because we know it's going to work every time. So highly recommend this one if you're running sulfate. And again, a one reagent test. You're using small amounts of this reagent on each sample as well. So this one is a very nice value for your dollar. And then silica is the next one, similar to the phosphate chemistry. This is for our EPA-121 method, going up to 25 milligrams of silicate per liter. We have 2 reagents here. It's a 3 reagent test in the method document. The 2 reagents we have pre-made are the oxalic acid and the ammonium molybdate. The third reagent that's used in our method is a 10% hydrochloric acid reagent. Inorganic Ventures did some studies and some statistics on this and determined that it's just not worth buying pre-made because it's so cost effective to make in-house. As long as you buy, the ACS grade hydrochloric acid, you're not having any issues with performance here. Super easy to make, super cheap to make in the lab, doesn't take much time. So rather than having that one pre-made, we suggest just making that one in the lab yourself. You can get the other 2 pre-made and still have some nice quick prep for your silica method. Coming soon, we have total hardness. This one is for our EPA-106 method, going up to 400 milligrams of calcium carbonate per liter. This is another one that I love having pre-made because the reagents for this one are a little bit funky. We've got a magnesium EDTA buffer that uses a whole bunch of ammonium hydroxide, which is just very, what's noxious? Is that the right word? The smell is very intense. Definitely want to be using a fume hood, but sometimes it's just nice to not have to deal with the fume hood and disposing of things in the fume hood, especially if your fume hoods are crowded or there's a lot of competition over who has the access to the fume hood. So nice one to have pre-made there, as well as the Calmagite reagent. Calmagite's a very intensely colored chemical. So having that pre-made is just nice to help prevent messes around the lab. And then, like I said, what's next? Let us know what tests you'd like to see pre-made reagents for. TKN, we've had a lot of requests for already. TP, total phosphorus methods. Again, we have the orthophosphate methods. The ascorbic acid from that can be used in some of the total phosphorus methods we have as well, but not all of them. So let us know which analytes, but then also which specific seal method numbers you'd like to see pre-made. Cyanide, phenolics, lots to hear about. So let us know your opinions. And that's all from me.
Thanks, Johan. I'm gonna try to figure out how to share my screen. I can't.
We're all learning here.
Okay. Are we rocking and rolling?
Oh yeah, we're good.
Okay. Very nice. I just wanted to briefly, we're kind of wrapping it up. I just wanted to briefly go over what resources we have available. So of course, Seal Analytical or Inorganic Ventures, our tech support teams for both companies are incredible. I know I go to Seal's tech support all the time. They're probably loath to see my phone number pop up on the thing, but Seal Analytical is a really good place to start if you're having issues with your methods or anything at all, and then we can help troubleshooting any issues you're having with reagents. And if you just have any general technical questions about the methods, the reagents, both companies are a great resource and happy to help and happy to answer questions. On the Inorganic Ventures website, we do have a product flyer and then some method sheets. So the flyer goes over which reagents are available, what methods they're similar to, what their names are, so you can use that as a reference when you're purchasing them. And then the brief description of what it is, if the name's not descriptive enough for you to be able to match that with whatever method you're running. And then the method sheets have, again, what methods it's similar to, the EPA or ISO or standard methods that it's like co-equivalent to. It has the reagent composition. There's usually some notes about, some standards you could use with these methods, the information about what chemistry is happening, what reaction is going on. For some of them, there's like tips and tricks, like specific maintenance requested, or things that you might see with those reagents. So the method sheets, I think, are a really valuable resource, and they're the seal method sheets that they give. It's just a little less information dense, and the tips and tricks listed in there are just a little easier to access without having 14 slides of everything else, or 14 pages of everything else to look through. I think they're really good, quick reference if you are having issues or if you are looking to get into running a method on your discrete analyzer. I think it's really easy to look at those and see, is this going to be feasible? Is this going to work for me? They're a really good reference. Like I said, the Inorganic Ventures R&D team has done a lot of work to develop these reagents. So every one of our R&D team is also, helping with technical support. So if you reach out, if you see our names, now you can know our faces as well. But like I said, both companies, CL Analytical and Inorganic Ventures, the tech support is, it's next level, and they're all so helpful and so willing to share information and help you figure out and troubleshoot. So really contacting anybody is a good bet, and you'll probably get the help that you need. So now we'll do some questions. I think Amanda's going to moderate the chat. Amanda, do you want me to leave this slide up? We have some, this is just some QR codes to some resources we have on our website. Not necessarily discrete analyzer reagent focused, just other stuff that we have.
Yeah, go ahead and leave that up. I also want to remind everyone that we've been posting some resources that we've linked throughout the presentation that you can access from there. And we do have several questions, so I'll go ahead and jump into those. So this question came in: which phosphate reagents were you testing? Were you testing both reagents together or each separately?
So that chart that I showed, the graph that had the calibration curves, that was both the ascorbic and the color reagent tested at the same time. So they were both, made the same day and then tested the same period for that. I think it was like 800 some days. So it's both reagents having the same, amount of stability testing happening to them. So they're both 833 days old and being tested to make that chart.
Okay, great. Thank you, Liv. Next question: can the 4500 SiO2C reagents be used for 4500 SiO2E method, which adds a reducing agent?
CO might be able to answer this a little easier, but if I'm remembering correctly, they're the same reagents. It just adds that reducing reagent. We had issues with long-term stability for the reducing reagent, so that's why we don't offer that one. But Johan, I think, might know if they're the exact same.
Yeah, they're actually a little bit different. So at this point, we only have the pre-made reagents for the 3 reagent test, not for the one with the reducing agent added in.
Okay. For ammonia EPA-153-C on the AQ400, our daily prep includes 3 grams of DCI and 14 grams of NaOH. Where the method only has 7.5 grams of NaOH. This was the sweet spot using the bucket test. How is that dealt with if we are using the pre-made reagent? Would you still recommend running bucket tests and adjust the NaOH if necessary, if needed?
Yeah, that's a great question, and I'm glad you asked that because I meant to talk about this on the ammonia slide, and I felt like I was forgetting something. So thank you for that. The ammonia reagents that we have made through Inorganic Ventures right now are for unacidified samples. If you look in the method document for ammonia, sometimes there's information about preparing for unacidified samples versus acidified samples. It sounds like you're using acidified samples and adding that sodium hydroxide into the DCI reagent to account for that extra acid in the samples, which is exactly what you should be doing. So these reagents are for unacidified. That way, if you are running acidified, you have the option to add in some sodium hydroxide to bring that reaction pH up into your necessary range. Most likely, exactly like the extra amount that you're adding now will be how much you need, but it would be good to do a bucket test, especially that first time, just to make sure that you're in the right range. And then you should have a pretty good new, baseline for what to do if you're using that pre-made DCI reagent. The shelf life that Inorganic Ventures has tested is for the exact recipe that they have. So they haven't tested it with that sodium hydroxide added in. So what I would do is wait until it's time for you to use that bottle, then add in the sodium hydroxide. With the size that the bottles are, you'll probably use up that reagent before you're even close to 9-month to 24-month expiration date that Inorganic Ventures has listed. So the slight change or possible change in shelf life shouldn't be an issue. But do keep that in mind that it technically hasn't been tested at that level yet. As we continue to get experience with these pre-made reagents and see what requests are out there, there may be an option for pre-made reagents for acidified samples for the ammonia method as well.
Great. Thank you, Johan. Another question, are the stabilities for the reagents listed on your websites? Our lab only runs the AQ300 a couple times a month and would love to save reagents and chemicals if there are options for reagents with greater stabilities.
So in that document that I mentioned, the flyer, the sales flyer that lists all the reagents, there should be expiration periods listed for each of those as well. And as we update, like I mentioned during the stability section, as we're doing additional testing and updating those expiration periods, if that data looks good, then we'll update the cert on our website. And so your documentation will be updated as well. So you can find that information before you purchase, and then if you're checking back on them, you'll see if it's been updated.
Great. Thank you, Liv. Also, the flyer Liv mentions is linked in the chat box. It is the first link that mentions our full line of reagents. So you can access that there. Next question, are the shelf lives for each reagent listed on the Inorganic Ventures website? If not, could they be? So yeah, we just answered that one. Thanks, everybody. Next question. Good morning, Liv. Is there currently a CRM for matrix water available?
I'm not sure, but you could certainly reach out to our custom product development team and they can let you know if we have a product that matches what you're looking for. And if not, we can help you design one.
Yeah, just send an email to quotes@inorganicventures.com and we'll have one of our custom product development chemists take a look at what you need specifically for sure and reach out to you. Great question. Next question, will larger volume reagents be available? That's a great question. I would say what volume, if you want to type in here, Larry, if you want to type in what volume you're looking for, or if you want to email one of us directly. I know some people have very high volume, high throughput, and it's something we've tossed around based off that, making larger volume sizes available. So if you want to reach out to us and let us know what your volume needs are, if it's not available, we can certainly explore a larger volume for you specifically, or anyone else that needs it. We're willing to be as flexible as you guys need. So you can email me, amiller@inorganicventures.com, or again, quotes@inorganicventures.com, and I'm happy to give you a call and talk about what you need. Okay, next question. Johan, we hope next year there will be a standard with matrix oil.
Can you repeat that one?
Yeah, we hope next year there will be a standard with matrix oil.
Matrix oil.
A standard like a CRM or like a method to run for that?
Yeah, if we could get a little more clarification on that one. We have lots of questions coming in. This is great, guys. Yeah, if we could get a little more clarification, are you looking for a CRM method or standard or a method involving matrix oil? Let us know specifically what you're looking for with that one.
Yeah, I apologize if that's based on a previous conversation we've had. I woke up very early this morning, so I'm a little foggy.
I don't think it is. I don't recognize what that would be as far as discrete analyzer interaction goes.
Okay. Next question, how about TKN reagents?
Yes, TKN has been by far the most popular request, and something that we're beginning to explore. So no promises on a launch date, but I think that we're all excited to start working with that idea.
Mm-hmm.
Great. Next question. I'm so glad to hear that cyanide is being considered. We are very pleased with Inorganic Ventures products. Did I understand that after the TCT bag is opened and stored refrigerated, that the product needs to be stored back in the TCT bag with binder clip or some other closure for preservation from light? Thank you in advance.
Yes, we do recommend protecting them all from light. So like I said, just popping it back in that bag, or if you have a container in your fridge where you can just store them all, not in the bag, but they'll be protected from light, like a sealed box or something, then that's fine too. Just whatever's, most convenient for your lab and your storage availability.
Okay, great. Next question, are pre-made reagents you have now available for TP analysis? Could you explain more about what methods they can be used for as of now? It seems you do not have a dedicated reagent for TP.
That's correct. We don't have a dedicated reagent for total phosphorus yet. The ascorbic acid reagent we have for the orthophosphate method, EPA-118, that is a 1.5 grams of ascorbic acid into 100 milliliters of DI water. And that ascorbic acid recipe is used in a couple of our other total phosphorus methods. So you can check your seal method document. If it says 1.5 grams per 100 mils, you could use that ascorbic acid. The color reagent is not the same. So that one we don't have available yet. So that's just kind of a, if you want to start working with pre-made ascorbic, you can. Otherwise, you can wait until we have dedicated total phosphorus methods or reagents for pre-made reagents for our methods.
Okay. Next question, to follow on the NH3 DCI question, are all of these reagents made for unacidified samples regardless of test?
Yes, these are all for unacidified, kind of the default reagents that are listed in our method documents. For most of these, it would be the same if they are acidified. For nitrate plus nitrite, it can vary method to method depending on the sample volume compared to the buffer volume. But if you look in section 11 of your method document, it will give you some tips on seeing if the buffer recipe as written will work with your acidified samples. You do a little pH comparison, mixing some sample and buffer together with acidified versus unacidified, and then see if that pH is within a certain range. The method document will tell you all the steps there. And there's a good chance that that buffer will work with your acidified samples as well.
Great, thank you.
Next. The buffer can handle up to like 5 mils per liter sulfuric acid, which most labs are not using that high a preservation.
It did change a little bit on some of the revisions we've made to, I think I can't remember if it's the lower range or the higher range, probably the lower range methods. We did some optimization that improved the performance of the method, but it meant that the buffer doesn't quite handle as much acid as it used to, but it still sometimes does handle as much as you need. So check that section 11 in your method document and it'll help you out.
Okay. Next question, why are the stabilities listed by seal for the prepared samples so different from the prepared samples by IV? Does this mean that the samples we prepare in our lab actually have a longer shelf life?
No.
The answer to that last part is no. Inorganic Ventures gets the business that they do for a good reason with some very impressive techniques that they use. And I don't think I can talk any more about that, but I don't know, Liv, if you want to add anything.
Yeah, kind of, I don't like the word proprietary, but we've done a lot of studies on these reagents and done a lot of testing to make sure that they're lasting as long as we're saying that they are. So, the ones that we've made and have tested and approved, I think, obviously we're giving it that shelf life because we have the data to support it. But for things that you're doing in-house, I don't think it has quite the same implications, I guess is the word.
Yeah, thank you both. Also, we'll look into why the stability is listed different between Seal's website and our website, because it should be the same. It should match. So
we're talking about stability of made in-house. Versus
I see.
No, we list the same for the pre-made reagents, but
Okay.
In our method document, when you're making it in-house, there's a much shorter shelf life.
Okay, gotcha. All right, thank you. Okay, would love to see a reagent set for hexavalent chromium in the future.
Duly noted.
Okay. Next question, once the reagent is poured into the wedge, does the expiration change or will the reagent still hold the original expiration time?
So I have noticed in my testing, one, probably not refrigerated as well in the wedge, because it's not in a refrigerator, not protected from light in the wedge if you're not keeping it dark. And also those wedges aren't airtight. So if you have like the hardness reagent that has all that ammonium hydroxide is really noxious and stinky, and the fumes get everywhere, and they can get into the other wedges if you have those sitting beside each other for a long time. So I've noticed that can affect. So I would recommend not leaving them in the wedge for a year and saying that that's fine, because it can be experiencing a lot of things that it's not going to be experiencing in a sealed bottle in the fridge in the dark.
Yeah.
That's once you pour it into the wedge, it's a whole different exposure to the elements into the atmosphere and evaporation, vaporization. So that's what kind of like if you pour a glass of milk and set it on the counter, it's not going to last as long as that gallon of milk in your fridge.
Great, thank you. Next question, is there a possibility for reagents for the AA500 in the future?
Is that a flow?
I'll leave that to the
So the
product
AA500 uses a much larger reagent volume. Not saying that we can't do it, but that does end up being more expensive for a lab. And so to validate that increased cost might be a little trickier, right? With discrete, a lot of these reagents we're using 100 microliters a sample, something quite small. So when you get the 125 milliliter bottle, you might have a couple thousand tests out of that. Whereas with a flow system, some of the reagents we're pulling, 1 mil per minute. If the system's on for 2 hours, that 125 mils is drained already. So it's a little trickier, but if there's demand for it, we're not against looking into it.
Yeah, I will say if you want to email me on that one, also, this is talking into volume, so certainly email me amiller@inorganicventures.com and let me know. Happy to set up a call to talk to you about that for sure. Next question for the nitrite method. Make sure I read that correctly. I did. Can the buffer solution be available without the titrin added for the nitrite?
without the triton added. Oh, specifically for nitrite testing.
Yeah.
That's something we can add to the request list for sure.
Okay.
So yeah, so for nitrite, we have a method for each range with and without the buffer. So it's possible also to run that method without utilizing a buffer and just use one reagent. The other option is, look, the Triton isn't going to help necessarily with nitrite, but you still should still be able to get pretty stinking near the MDLs that we have using a little bit of Triton in that buffer itself.
Yeah, that's a good point. If you're not trying to push for the absolute lowest MDL possible on that method, which is pretty darn low, you can probably leave the Triton in there and get perfectly good results.
Great, thank you. Next question, should the phosphate spike be added to the ascorbic acid at the same ratio as it is if making the reagent from dry ingredients?
Same ratio, yes. Same exact volume that's listed in the method document. Probably not, because in the method document you're making 100 mils, and this is coming in a 125 mil bottle. So you have to do the old C1V1 equals C2V2 when you're preparing that spike. But yeah, the same ratio, correct.
Great, thank you. Next question, where do we send our method recommendations?
I would send that to, I'll put it in the chat, inform
Okay.
at seal-us.com, unless you wanted to go to Inorganic Ventures.
Yeah, probably send it to Liv also. Liv,
Oh, okay, that sounds good too.
Yeah, you want to pop your email address in the chat also, Liv?
Yeah, I can put my.
Oh, there we go.
Yeah. I'll put my individual email in the chat. It's just Forbes, Forbes @inorganicventures.com. Actually, since I'm sharing, can someone do that for me, please?
I gotcha.
Thank you.
Thank you. Next, looks like we have a request for ammonia chloride buffer. So, okay, do that one.
So That should be the nitrate buffer,
Okay.
depending
right.
on what concentration you want. I mean,
Yeah. No. Lorraine, if you have more details you want to share on that would be great. Okay. And then the next question, since we should not store them in the wedge after one run, how much reagent are you pouring up per run? And is that still poured in a wedge first, trying to decide how long the 125 ml reagent bottle would last?
Sure, I see what you're getting at. That's going to depend on how many samples per run that you're doing. You can check the method document and see how much reagent per sample is being used. Multiply that by your samples, calibration standards, controls. See how many pulls out of that bottle you're doing per run to get an idea of how full you want to fill that reagent for each run. You don't necessarily have to report after each run. But I generally don't like to leave my reagents sitting on there for more than a day. You know, some labs do leave them on there for like a week at a time, depending if you're trying to hit low MDLs, it's probably not the best idea. If you're just trying to get some really basic results and you're not too concerned about the lowest detection limits or the lowest precision, then maybe you might be able to leave those on there a little bit longer. It's kind of a lab to lab basis thing. It's hard for us to give an exact answer on how long is okay to leave that on your system. But for best results, definitely at least pour up fresh each day. If you're running low level ammonia, then maybe you do want to repour each run because of that ammonia in the air that might be contaminating your reagents over time.
Hey.
Yeah, but we also don't want to scare you, right? So if you've been running with making up the reagents in-house and you have them in the reagent container, right? We do supply closed caps. We do supply on the system. The reagent tray does come out and goes into the refrigerator, right? That's all by design. So if you've been doing it that way with the reagents that you've been making up in the lab, you can absolutely continue to do that with these reagents, right? When they come out of the bag, they magically don't all of a sudden stop working within 24 hours. So don't be worried about that. You can probably continue to use them as you have been with regards to storage. Just make sure that the rest of that bottle is still in that bag and sealed so that we don't have that being exposed to light.
Yeah, great point.
Mm-hmm.
And then last question I see here, does the pre-made NEDD turn pink? And if so, would it still be usable if filtered?
It does turn pink after nitrite touches it, right? That's the
It should.
so it
don't-
might have a slight tinge to it over time, and just like is recommended in our method, you can filter that. That actually creates a pretty big molecule that can usually be filtered out a little bit. So absolutely, you can try that if you have that and had that reagent opened and it's been sitting around for a while and it's just got a little pink tint to it, just like it says in our method, go ahead and pour that through a tight filter, and then you can use that reagent on the system.
Yeah, Liv, I'm curious, have you noticed or do you remember if you've seen that NED reagent turning pink over time in your testing?
I know the room temperature one definitely has. I'm pretty sure the refrigerated one looks pretty good still. I mean, like you said, a slight tint, but we filter it and it's fine.
Nice.
Okay, one more question. Chloride combined color reagent needs to be filtered prior to use if it sits around for a while. Does the pre-made need to be filtered also?
We haven't seen issues with it having any sort of particulate or anything in there. If you see it in there, I would recommend filtering it, but like I said, we haven't had any issues. We filter them before, like while we're packaging it, so it should be good to go.
All right, very good. I am not seeing any other questions pop in here. So I think we are good. So, and we are at time, we're a little over. So thank you everyone for your patience. Thanks for all the great questions. If you have any additional questions, certainly reach out to anyone with the emails provided in the chat or on this slide. And we hope to see you all supporting this effort and purchasing the Seal Analytical pre-made reagents to save your lab's time and money. So thank you again, everyone, for joining us, and we hope you have a great day.
Bye.
Take care.
Thanks,
Nice to
guys.
see you.
Bye, everybody.